In 6.4, we will talk about ATP or adenosine triphosphate, which is our short-term energy storage molecule, which is very important in our cellular respirations and all the biological processes. As you know, we talked about ATP a lot before, but it is very small and simple molecules that basically powers the majority of the energy requiring cellular reactions. And the structure of ATP that you can see here, it's basically have an adenosine, which is a nucleotide. It is a DNA nucleotide. It is actually a RNA nucleotide because this is a ribose sugar. It's not a deoxyribose, as we remind yourself from chapter 2, and it has a nitrogenous base. So it's that's base A, and it has a 3-phosphate group, and we will name that first phosphate group as an alpha phosphate group, the second one will be beta, and the third one will be gamma. So alpha, beta, gamma, first, second, and third phosphate group. So remember when we say we're forming bonds, we will store energy within that bonds. So that bonds that we're seeing right here is the bonds between these two phosphate and have oxygen in between. That bond will store a lot of energy that will allow you to extract, to do work when we go through the process of ATP hydrolysis. So hydrolysis here is you have to use water to break that phosphate apart from the rest of the ATP molecules. So when we take water, combine with that with ATP, we will have ADP, or adenosine diphosphate, meaning 2-phosphate group, and you get an inorganic phosphate. That I stand for inorganic, and you have energy being released. So again, delta G naught is because this is at a standard conditions. It's negative is because it releasing energy. So delta G naught for this particular reaction, the energy release, the free energy release is available to do work, is going to be negative 30.5 kilojoule per mole. So all of this is per mole of ATP. You will have you will release 30.5 kilojoules. So joules is the unit that we use to depict heat or Gibbs free energy being produced in a certain chemical reactions. You can further break down ADP into AMP, or adenosine monophosphate, and another inorganic phosphate, and that will release another 45.6 kilojoules per mole of ATP. And that is between the alpha phosphate or the beta phosphate that you see here. So that's the first and second phosphate for it to become ADP, for AMP to form from ADP. And if you break the third phosphate, you will form ADP from ATP. And all of this, we call the ATP hydrolysis. Remember, we have all of our reaction before hydrolysis. You require an addition of water in order to break that bond. You also see this reactions in the middle, this chemical equation in the middle, showing you the exact same as the above, except this is not at a standard conditions. It is in your cell. So if you're having ATP hydrolysis within your cell, you're actually releasing a little bit more of energy as opposed to the standard condition. So we're a bit more efficient at extracting ATP. And these bonds that we're seeing right here between the alpha and the beta and the gamma, those are considered a really high energy containing bonds. And this process is reversible because we know that we can use ATP synthase to make ATP from ADP and an addition of an organic phosphate. And of course, that process will require an input of energy in order to form that ATP. ATP overall is a really highly unstable molecule. So if we leave ATP alone, it will just spontaneously dissociate into ADP and an inorganic phosphate. So when we produce ATP, we should use it up as soon as possible, or else all of our energy input will go to waste, and the free energy released during this association process will be lost as heat. So in our cells, we actually use this ATP hydrolysis and couples that with another endergonic process, or a process that requires an input of energy in order to perform work. So this is your friendly sodium potassium pump that you've seen before. So our NAK plus pump that will export 3 sodium ion outside of the cells and import 2 potassium ion into the cells. That process is an endergonic process. If you couple that or combining that with the ATP hydrolysis or an exergonic process, whereas this process releasing energy and this released energy will be used to drive the other endergonic process and allow you to maintain a stable cellular concentration of Na plus being more outside of the cells, and there's more K plus inside of the cells. So at the bottom line of this energy coupling is you couple a energy requiring endergonic reactions with an energy producing exergonic reactions. And for most of our cellular processes inside our body, that will be ATP hydrolysis. Another example that you will see that is relevant to our chapter 7 will be glucose conversion to fructose 6-phosphate, which is the very first step of glycolysis. For fructose 6-phosphate is a unstable intermediate where requires that ATP for the conversions from glucose to fructose 6-phosphate, and we want that unstable intermediate to drive further reaction down the line of glycolysis. You will see that in chapter 7. And so again, you see that exergonic process of hydrolyzing an ATP and an endergonic process of this conversion, and you're coupling that together to create a reactions where you're using energy from one process and use it for the other process that requiring energy. So this is just the 6 steps that you've seen before in lecture 5, where you have your sodium and potassium pump. Whereas here we also put in some details in regards to energy and our phosphate group. So first you have your sodium potassium pump in a 1 conformations where they will open toward the interior of the cells. That forms without the phosphate group attached. They will have high affinity for sodium ions, thus they will bind 3 sodium ions. This binding will trigger the pump being phosphorylated. What that means is the ATP will hydrolyze and transfer that phosphate group, the gamma phosphate group, to the pump and changing the shape or the conformations of that pump, cause them to open to the exterior of the cells and export that Na out of the cells. After the Na left, they don't have a high affinity for Na plus anymore. They will actually now have high affinity for potassium ion or K+, and they will bind to 2 potassium ion. When they bind to that potassium ion, they will be dephosphorylated. What that means is the phosphate group will leave the pump, cause them to once more time change conformations and now open toward the inside of the cells. Then, because of that conformational change, they will have a low affinity for potassium ion, and they will release that potassium ion and switch affinity to the sodium ion, and the whole process start again. So this process will be very important to maintain your cellular salt concentrations, and just to show you that you can use ATP hydrolysis to drive an endergonic or an energy requiring process. Here we have a sample problems where you will see again possibly on your test. So if you have 1 ATP molecules, 1 hydrolysis will release 7.3 kilocalorie per mole of energy. Your delta G for that process is negative 7.3 kilocalorie per mole of energies. So if it takes 2.1 kilocalorie per mole of energy to move 1 sodium ion across the membrane, so this process will require energy, so you see this plus sign. You require the input of energy, whereas this process above ATP hydrolysis release that energy and produce 7.3 kilocalorie. So how many sodium ion can you shuttle across the membrane? So you can just simply do just to count them. So one molecule requires 2.1, two molecules will require 4.2, and so on and so forth. You go all the way to 4, 8.4. And at this point, oh, please ignore that one. At this point, it is higher than your 7.3 releasing energy. So you can safely conclude that you will be able to transport 3 whole Na across the membrane. Or you can actually do a simple math operations to get the question answer as well. You can take 7.3 divided by 2.1, you get 3.47 Na plus ion, but you cannot transfer 4.7 sodium ion, so you only transfer 3 across. So I would recommend you do this quick way during the test so that you can obtain your correct answer. And that concludes our 6.4.